Thank you for being here. We're going to have fun. Many of you are already familiar with the Zuckerberg initiative, but you're going to learn a lot more about that today. We are about 8 years into the work that Priscilla and Steve and others have started, and so we're going to talk about AI, and we're also going to talk about the way the Zuckerberg initiative thinks about big ideas. But because all of you are humans, and humans love stories, we're going to map this onto the story of the Zuckerberg initiative, kind of its birth and early days, and now it's sort of formative years that we're in right now. And so I want to go back to the very beginning, even before CZI launched. Okay. The two of you on the spectrum of sort of how people are situated are unusually strong believers in the transformative potential of technology, not only in science, but elsewhere in society. So I want to hear from you a little bit about how did you become so convinced that technology is the right thing to work on where there are other alternatives?
Good morning, everyone. Happy Saturday. Thank you for being here. For me, I started my journey to wanting to take care of kids. I was a school teacher and then trained as a pediatrician at UCSF. And as many of you know, when you work in a tertiary care center, a lot of the kids come in have illnesses that don't have names for the diagnosis. They don't have known treatment plans. You're really digging into the research and trying to use the research to find answers for patients. And I have a very clear visual in my mind of a parent holding like a PDF printout and being like, this is the hope. This is what I have. And me as a medical student, intern, resident would be like, I don't know what this is. Like, I got to read through it. But basic science research has really come to me represent hope and represent hope for patients. And Mark and I would talk about this, and at the time still, like, he was deeply embedded in building technology at Facebook, now Meta, and I was really thinking about how can we improve research, see the world in new ways for kids and for research. And that's how we started thinking, like, how can these ideas come together, and also inspired to think about how we can build tools. Because if we can build new tools to help scientists move faster, think differently, see the world differently, like, that shapes the way you think about solutions and possibilities in the world. Steve and I love the story of the microscope and how the microscope transformed the way that we think about human health and introduced the idea of microbiology. And so we really felt like, what are the types of tools that that CZI can help build to accelerate the whole field in being able to make progress towards the goal of cure, prevent, and manage all disease. But I have a story about that I'd like to tell.
Tell me. Tell us.
So we didn't look, it's Mark and I were the ones who were like, look, we want to set an audacious goal. We want to be part of cure, prevent, and manage all disease. We started with cure. Corey Bargman convinced us to add prevent and manage, and we're like, well, that's a mouthful, and can't you just cure? And she was like, well, like, if we add this nuance, I think there might be a way. And okay, so Corey, that was Corey's contribution, and that really set us in thinking about, like, people often ask, like, okay, which disease do you start with? But that's not the point. The point is, how do we think about the entire scientific enterprise, and how do we make basic science research faster, more effective, see the world differently? And so that actually changes the entire strategy. So when you set these audacious goals and get great scientists to think about it, they can get to a point where the strategy can actually get you there. But Steve Quake was very uncomfortable with this for about a year. Steve really wanted to do great science. He was excited about it. He was excited about the long-term mission. And we launched in September of 2016. And we were at UCSF then. We were on this big stage. And, like, the only coaching Steve got was, don't laugh when people say cure, prevent, and manage all disease.
I
couldn't say it with a straight face.
No.
Right?
We
I
would
just
be
could
like,
not. To
no.
you or Mark either. Right?
No.
I mean, I don't know why you hired me because I couldn't say the mission.
He really couldn't. We'd be like, okay, just like keep a straight face.
Can I ask you about that? Because when you said that, I'll invoke the grumpy scientist. There were a lot of grumpy scientists who were like skeptical. Yeah.
Skeptical, please.
There's no grumpy scientists.
These people were saying you're being unrealistic.
Yeah.
And what I recall was that part of the argument in favor was that the rate of technology advances, the rate of these advances is increasing over time. The gap between them is getting shorter and shorter. And that was an interesting argument. This was all long before AI. So do you feel, how do you feel now about setting that audacious goal now that everybody is sort of worshiping at the altar of AI and nobody realized that was going to happen before?
Well, Steve and I gave each other a high five when there was one like previously grumpy or skeptical journalist who wrote a tiny article that said, maybe they were right. Maybe it's possible. And I was like, yes.
People aren't used to thinking about progress on a 100-year time scale. I mean, they're not. I mean, maybe historians are, but not scientists. And, we think on grant level 3-year timescales or PhD level timescales, 5 years, or 10-year level timescales. But think about, we'll talk about a decade in a minute.
Yes,
and even that's a stretch, but 100 years, nobody does. And I think part of the initial reaction of skeptical people, they're just not used to thinking about what happens over the course of 100 years. And when you get kind of scholarly about that, you realize a whole lot can happen in 100 years.
And when one of those advances is artificial intelligence, and that alone, especially when mixed with the other things that people are putting together, the potential is outrageous. So let me go back to something you just said, which is timescales. The Chan Zuckerberg Initiative is a super compelling organization and unusual in the sense that it doesn't target its funding, as you said, to one disease or to one area. You think in terms of, apropos of the topic of this conference, very big ideas, ambitious ideas, and you think on a 10-year timescale. Can you explain why the 10-year timescale is sort of the right sort of segment of thinking, the right sort of way to measure a truly ambitious idea?
For us, 10 years is enough to really say, I want to do something big, but not long enough where you can just hand wave the whole time.
Yeah.
You have to be concrete. You have to say, build a plan. You can have open questions that are unanswered. You should. But you can concretely say, I'm going to start here and I'll learn something new. I'll do the next step. And it also allows us to think about like, okay, well, have we achieved something at the end? We've actually remarkably been around for almost 10 years. And you can say like, has this chapter been successful? Evaluate, decide what you've been good at, what you haven't been good at, and set the next 10-year goal. And Steve's been part of the CZI world since our inception, but moving into leading all of our science work, we've really started thinking about our different levers at CZI. Do you want to talk about like build, do, fund?
Yeah, sure. And it's a very unusual philanthropy in the sense of having these enormous ambitions at 10-year timescales. And the strategy for going about it, build, fund, do. Fund is grant giving, which we do internationally in something like 34 countries around the world. And that lets us reach out and connect to great scientists anywhere they may be and support their work. Do is doing science, which is starting institutes, labs, and having our own staff do research. And that was my first entree into the CZ world as the sort of co-founder and leader of the San Francisco Biohub. And then build is building open source software to accelerate science, which I don't know if any other organization is really doing that, certainly not the scale we are. And that's been sort of one of our secret weapons.
Let's talk about one of those 10-year timescale projects that you started with, which is also a way for me to get us into talking about AI. And that was a project to catalog all of the cell types in the bodies of humans and other animals. Anybody in the audience who's taken a course in cell biology might think, wait a minute, I thought we already knew all the cell types. So explain why that was a compelling 10-year idea and how that worked and what you found.
Yeah. Well, I would say it's one of the great achievements of the last 20 years to sequence the genome, the human genome, lots of other genomes. And people talk about that loosely. They say, well, the reason we did that is the genome is the blueprint of the organism. But that's not really true, right? 'Cause pretty much every cell in your body has got the same genome, and yet they're quite different. Heart cell different than a lung cell, different than a brain cell, neuron, whatever. I mean, even though they got the same genome, how do they become different? And how can you predict the cell types of an organism, of your body, from the genome? You can't. So there's like this huge missing conceptual fundamental piece of how genome goes to organism. And without a principled way to do that theoretically or computationally, we decided to resort to experimental methods and try to build molecular characterizations of all the cell types in the body. And that builds on technology that I was involved in developing early in my career. And sort of my reasons for getting involved was, I wanted to do this really big project that I knew was going to be a team science project well beyond what my lab or any single lab could do. And we set up the Biohub to help take that on. And in parallel, CZI set up grant programs to help take that on, to energize it around the world. And collectively, it was like the first big bet, I think, of your science philanthropy. And it's really, really paid off.
Priscilla, what was your experience of seeing this project, kind of unfold? It was a huge bet in terms of people, money, and ambition, and it was hard to know what you're going to find. How was it sort of sitting in the in the front seat watching this happen?
So
I got a story. Wait a minute.
Okay.
Let me answer
I
that
don't know where this is going.
So we took parallel approaches between Biohub and the grant program. Grant program was funding deep dives in single tissues in different groups. And Biohub, we were like, we're going to do the whole thing. We're going to do all the tissues from one organism, 24 tissues, and try to figure that out. And so we started with mouse. And Tony Whisker was a great, sort of partner in that. We decided we're not just looking at mouse, we're looking at mouse aging. And so we did that to kind of warm up. When we started the Biohub, I didn't tell you that, but we had no idea how to do it. And we just said, we're going to figure it out as we go. We know the basic technology is ready for it, but there's a lot we got to figure out. And so we decided to do mouse first. And we did, and we learned a lot about how to both technically do it, do the team aspects of it. And we were updating Mark and Priscilla in the board meetings. And at one point we said, well, we got it. It's a rough draft. It's ready to go. And Priscilla said, well, why don't you just do human? And that set us on the path. You remember that, right?
Well, why did you say that?
Well, I was ignorant. I mean, truly, sort of. I mean, like, I was an MD. I knew enough to know, sort of, to ask questions, but I don't, know the complexities of doing basic science research. And so I'm like, well, that's great, but haven't we cured diabetes in mice so many times? Can we work on humans? And I remember that was a complex project.
How did it work?
Tabula Sapiens.
Yeah.
But I think also it was like a lot of people in the field were thinking about how to do human. There's a human cell atlas project that was very diffuse. And it was like, I think people felt it was like a 20, 30-year goal. And having done mouse in just a few years for not a lot of money relative to what people were thinking, it was like, okay, maybe human's closer than we thought, and we should just go for it. That's how I interpret it, and we did with the Tabula Sapiens.
Totally. I mean, I think a lot of times my role and Mark's role is just like asking questions that sometimes in the science community is like, no, that's too hard, and we're like, no, that seems like what you would need to do to get to the next step. And so, that's
the push is catalytic.
Yeah.
And what did you find? How many cell types are there? I want to give people a sense of the scale of the data, because this leads right into where you were when AI came on the scene.
Yeah.
And the data you were holding. Yeah.
Yeah, and so, in our Tabula Sapiens, we've got about 1 million cells we've analyzed, and it's like upwards of 600-700 cell types, which is maybe double the number that's in the textbooks when we started. And there's a very interesting sociology of science thing about this. I'll take a little detour into that.
Please.
When we started, we had a meeting here at Stanford. It was like the first big human cell atlas community meeting, and I was the organizer. It was the second, I mean, the first big one. There was a smaller one in London before that. And here we invited a much larger set of people, and I said, everybody gets to talk. We're going to have like 3-minute talks. Everybody gets to get up, say their thing. Corey had just started. Mm-hmm. She was like so happy sitting in the back taking notes, everyone auditioning for her. And as an icebreaker, I had said, everybody on your name badge, write down how many cell types you think the human body has. And I was shocked by the lack of imagination of my colleagues. I mean, many of them didn't want to play the game. But the ones who did, numbers were all over the place, from 100 to 1 million to uncountable. I mean, there was no consensus in the field what the number would be. And people who were writing grants and trying to raise big money to do that were making claims about lots of different cell types. The numbers are huge. And I feel like we should do a little retrospective now of the literature, because there's been so much published. And in every single paper in this field, people say, I've discovered all the cell types that already exist, and maybe I found 1 or 2 more. So we barely doubled the number of known cell types overall. I mean, the one exception is the brain. That's like a special case. But for every other tissue in the body, we've maybe doubled the number. And the real value of this has been not the number of cell types, but the fact that we're doing a very deep molecular characterization of each one. So many of these cell types, we only knew like 1 or 2 genes to characterize them. Now we know thousands. And that's the real value of the endeavor.
But this project was really helpful in clarifying like what CZI's role is, because when we started in 2016, we're like, we want to help scientists do great things. Like that's a pretty broad like mandate, and we tried lots of things, but the single cell work really like showed us the power of the biohubs. Like how can we push through big projects, coordinate across teams, across universities, in like big team science type of way. But the broader international grant community played a huge role too. We gave grants to folks all across the globe to contribute to in building that single cell atlas. And what CZI did is we built a lot of tools, by the way, and a lot of them that kind of took off, didn't work. We built all sorts of things. And the thing that worked here was super interesting. It's a tool called CELLxGENE.
It's amazing. You should, if you're in the business, you should try it.
And the very first time I think Phil Smoot described it to me, is like it helps people look at their data and circle things. Yeah. And I was like, circle things? I was thinking like big platforms, and it was like this very simple and useful tool for scientists. It wasn't like it didn't promise to change the world. It promised to help scientists. And what happened is that it was actually useful to scientists.
We could have done Tabula Sapiens without it.
Yeah. And scientists started using it. Community was building around this tool. Like data standards were building around this tool. And it was so useful in this community, and it helped us bring together a group of people in a body of work that now has resulted in CELLxGENE Census, which is the biggest repository. It's open source, openly available, but the biggest repository of single cell data. And so the cool thing is like, how did this simple software tool help unify a community and build a large data repository that now everyone can build off of and use? And for that was like, okay, that's the power of software that we can build. That's the power of the Biohub. How do we bring together the broader community with our grant funding?
Oh, amazing. And now you've set it up perfectly, because you're sitting there with this unique and ultra powerful data set, and AI comes on the scene. So I want to hear a little bit about the way you guys thought about the tools that first arrived: language models, protein folding and protein design, and then use that to help explain your philosophy of what CZI should be doing in this area.
So, the first big bet around making cell atlases experimentally, I thought was a very successful thing for CZI. And when I got to take the role of head of science, I had a decision to make. Do we want to keep doing this or do something different? And I felt like the role of philanthropy is to do the risky, unproven things. And I felt in the world of cell atlases, we'd done the proof, and lots of other funders coming in, the field was off and going. So time for us to change direction. And that was a little hard message to deliver. And I sort of told the team, that was a big bet, it was successful, take a lot of credit for it. We're gonna make another big bet, but I don't know what it is. We're gonna do small experiments and funding and things for the next couple of years, and an opportunity will present itself to us. I was confident something would happen. And
Steve's always confident.
Steve is always confident. Thank you for saying that.
Yes. And sure enough, the LLM revolution happened, and we realized that we were incredibly well positioned to play a role in that. Not just because we had built this huge open source data set, not just because we had a founder who was deeply into it and one of the world's experts, but our sort of meshing of partnerships with great universities, having a tech team on staff, and being able to be nimble in our grant funding, we thought would put us in a great position to be kind of the philanthropic scientific leaders in a field that was going to be a challenging one to address because it's so expensive that much of the foundational work was being done in industry because the calculations were so expensive. And we couldn't match that, but we knew that we could bring resources that few other organizations could to try to do things for basic science open research.
Yeah. On this one, the other unreasonably audacious person in my life, Mark Zuckerberg, we were like, could we build a small, we'll build the biggest cluster, bigger than most labs can run. We'll start with 250 GPUs. And he was like, that just doesn't sound good. Right? He was literally like, if we're gonna do something, we need can we do more than 1,000? And I was like, I tried to do the bottom up analysis. What do we need? Let's build to what we need. And he was like, no, you need to build to what will challenge people to think differently about what's possible with the data and think differently about what types of projects we should and could be taking on. And so that was a new learning for me too, putting together the cluster, and now we have 1,000 GPU cluster that we're using to run and support a lot of the models that we're building. And it is expensive. How much were we spending on it every day? You just told
starting at the end of this month, we'll be spending $50,000 a day.
Yeah.
On compute.
It better be useful.
Yeah. I think it will be.
Let's talk about how it will be useful. Either of you could help cue up the idea that you're not doing the same thing that other people are doing. You've decided to do a new thing, and the new thing is that you're going to, in various ways, model the cell. Why is that an interesting thing to do? What is entailed in that, and what's going to be possible?
So. It was clear to us that the study of protein structure is being revolutionized by these approaches, and the hurdle of the sort of enabling discovery had happened, and great things were going to proceed, and so therefore we didn't need to do that. And then we thought, what's the next level of complexity to study? The cell. Our sort of mission, our North Star of the next decade is understanding the mysteries of the cell, and we just thought we were very well positioned to make inroads in that, both because of having built the data sets, but also being willing to take the risk of building models where people hadn't really started yet, and trying to pioneer that area. Where I think it's going to go is it's going to help transform biology from being 90% experimental and 10% computational to be the other way around. We're going to help build a set of tools that will have really powerful predictive behavior about cells. And most of the work will be done there, and you'll be doing a smaller fraction of your time on experiments. And you can think of examples of how many mutations do you have to add to make a cell turn into a tumor or cancer, and which ones will do that. Right now, you do that experimentally, and it's painful, right? And there's lots of models and cell lines and animals. But if you could predict that, and really just be very specific about the experiment, that'd be really a game changer. If you could look at a developing organism and say, if I ablate this cell or that cell, what happens in development, and predict that, it would really narrow the number of experiments you'd want to do to understand development. Or if you could say, here's a novel virus, what's it going to do when it infects a cell, and how is it going to change what's going on, and predict that. All that is going to happen within the next decade, and the tools we're making are going to help enable that.
And sort of my hope where that goes is really starting to think about precision medicine in a different way too, right? If we go back to the patient that is dependent on you reading this one PDF to understand their illness, I think we also know a lot of patients that do the genetic testing, and they get sometimes they get clarity, but most of the time they get what I think is the murkiest answer on this. It's a variant of unknown significance. And you're like, okay, well, there is something. What does it do? How does it impact me? And like, what if you could take that variant and actually look at it through one of these virtual models and say, okay, this is the protein that's misfolded. This is how it affects your liver, and these are potential downstream targets or drugs or therapies. Like, that's huge, both for the patients with rare disease and all these variants that we're seeing now that we're doing a lot of sequencing in patients. But I actually think we'll end up continuing down the road of understanding that common diseases actually should be broken down this way, like hypertension, depression. Like, the fact that we treat depression with try this drug first, see how it goes, come back to me. If it doesn't work, let's try this other drug, like, that maybe may work or may not work. Like, we just have no understanding of the true biology and molecular basis of things that affect a ton of people. And we waste a lot of time, and it affects quality of life, and sometimes morbidity and mortality, in a way that if we truly understand the biology behind both rare and common disease, we could treat it so much faster, and these models can help us get there.
It's amazing. Maybe take one example, the universal cell embedding that you've been working on, kind of an early example of what could be done in this area, and give people a sense of where does the data come from, how does the crunching work, and what does the work product look like? And then I want to ask Priscilla, sort of the CZI is such an amazing track record, not only of generating big discoveries, but making them useful to people, and maybe we could talk about how they could be useful not only to researchers, but also to physicians.
Yeah. So the universal cell embedding was our first big swing at building a model on the cell by gene corpus. And the idea was to build a large language model around single cell transcriptome data. So all the genes being expressed in any given cell, could you build a model around that would tell you something about the biology of how cells work? And so and this was work done by myself with Yuri Leskovich and a couple of very talented grad students here at Stanford. And it was sort of interesting. I mean, we trained it, we were careful not to teach it any biology. We wanted to see if it could deduce biology from just a matrix of numbers. So we really tried to train it with just, here's a matrix with a bunch of numbers in it, and the rows are cells and the columns are genes, although we didn't know that, but we knew that, and said, build a model on that and let's see what comes out. Because we knew a lot of biology, and we just then used that to interrogate the model. For example, we'd annotated all the cells by humans and looked at gene expression. We knew which cells were which and which genes were expressed where. And we were just amazed to discover all this emergent behavior in the model. We withheld data that we didn't train it on. So with Tabula Sapiens, we had a published data set and the second generation unpublished. We withheld the second generation, and we then asked, okay, having trained the model and been in embedding space, this data is never seen before, where does it go? And it all went into the right spot. So it had learned, the molecular properties of human cell types just from those numbers. So that was good. We asked it questions about cell type relationships. So we understand lineage relationships, like immune cells belong together, other cells development related belong together. That is all known. None of that was in the training. And when you ask in the embedding space, what's the distance between cell types that are close in lineage versus far in developmental lineage, that's reflected in the embedding space. So we had learned something about lineage without being taught that. We designed it to be able to work on different species, and we trained it on something like 8 species. And then we began to show it data from species it wasn't trained on. Ask, where does that go? And it managed to put that in the right space, more or less. And so it was learning something useful beyond any given species about the evolutionary history and relationship of cell types. And so it's not perfect. I'm not going to overplay it. And if you're interested in the details, it's up on bioRxiv. It's been there for a while, so you can see in sort of more specific detail how it's working. But it was so encouraging to me, and just like amazing, this idea of emergent behavior it was there in this model on this molecular data, and it gave me a lot of the confidence to say, let's make a huge bet in this space. There's a there, and there's going to be a lot of great things that come out of this.
Priscilla, you mentioned the sort of physician experience, which I think is an interesting anchor here, that you go into the room with the patient, maybe as a pediatrician, you don't have very much time, you order a couple of tests, you get sparse data. How could having eventually, not this model maybe, but its successors in the background to plug data into, transform the experience of being a physician or being the patient?
Yeah, I think it's critical for physicians to be able to quickly understand, like, what, like, you don't remember all your, some of you might have it on the top of your head, but like, I don't remember the basic biochemistry of what's happening and what this pathway affects. And like how to put things in context for a physician to use is so important. And at CZI, we really call this like staying close to the work, or making sure we have important biological drivers in a lot of the basic science we do, to make sure that it's not a interesting but esoteric body of work, but it's really applied and useful. And there's a couple ways that we do that. One is we do this work is open source and openly available. We want other people to interrogate it, use it to build something different or adjacent or contribute. And that's a huge part of our strategy, because we want to make sure it's continuously relevant to work that's happening everywhere across the scientific community. And we get feedback that makes it better. Another way that we do this is making sure that we ask good biological driver questions. Like our Imaging Institute in Redwood City, we have very, very cool microscopes. We got to make sure we're looking at important things for human biology. And then we have a portfolio at CZI called Rare as One, where we work with rare disease groups. And so, these are usually patients or parents of patients who are like, they can't stand for the lack of progress in the area that is affecting them the most. And they are passionate, powerful advocates that understand the importance of research. Oftentimes, it's not even going to be for their own benefit. It's going to be for someone else. But they are motivated to close the gap between research and outcomes for patients. And oftentimes, the distance is the smallest between what happens in the lab and what affects a patient. So those patient groups are constantly pushing to make sure research is relevant. They have their own research agendas that they've worked with advisors to pull together. And now we've started connecting those groups to our single cell portfolio, which is super cool and powerful.
This is already compelling, but now I want you guys to think forward to a point in time. Maybe I'll bring it back to the core mission of CZI, that like, let's leave the Corey part out. Let's cure all of disease. How would the models that we build 6, 7, 8 years from now be empowering people who are trying to come up with cures for diseases that right now are completely unmanageable? What is the process of somebody who's a drug discoverer or developer, or somebody who's looking for the root causes of disease? How is that different given the existence of something that you guys build?
Yeah, well, to follow up on Priscilla's last example, if somebody has a mutation in their genome, and it's a new mutation, a variant of unknown significance, what's the biology behind that? Right now, we don't really have a way to predict that. You can put it in animal models and see what happens. That's laborious, expensive. But the computational tools and the virtual cell AI models will sort of enable, I think, much better predictions of what is the potential significance of this, what's it going to affect, which organ systems, which tissues, which cell types, what's going to go wrong. We'll be able to do that all computationally, and that'll be super powerful. I think we'll be able to ask questions. If I drug this protein or this pathway, what's the consequence going to be in the tissue of interest and other tissues of the body?
Because most drugs fail from an unexpected toxicity. Right?
Exactly.
Yeah.
And people are already using the experimental cell atlas data for that. Look for on-target toxicity in other tissues and cell types. But we'll take that to another level. I think it's going to be a really powerful computational tool to predict that. And I think when you think about future therapeutic modalities like cell therapies, we're at the dawn of that as an era, and these tools are going to really revolutionize that.
Priscilla, how do you see the the role of these models just sort of against the backdrop of the core mission of CZI?
Yeah, usually my role at CZI is like roadmaps, milestones, timelines. But for me, the way that these models are going to help is just like unleash the creativity of people who pursue science. People in this room, like, how can we allow you to be your most creative self? How can we de-risk things, make things faster so that you can pursue your wildest ideas instead of your safest ideas? Because the cost of time and resources is so much lower that you can ask more audacious questions, and that's what's gonna save all of us.
Then to wrap up, since we're almost at time, let's end with something fun. We heard about this argument that the rate of technology advances is increasing, the time between them is decreasing, and we've just seen AI hit the scene, and it's going to be here forever. But what's something you're excited about that would come after AI? What's something that could potentially be even more compelling in the future? That's a hard one, I'm sorry.
You want me to?
You can go.
I mean,
give me a second.
Yeah. I just think we're going to learn so much about the evolution of life on Earth.
Great.
Yeah. I mean, one of the things I'm really interested in with these cell embedding models and everything we're doing about cell type is understanding the origin of multicellularity, right? And I think these are going to get us to a deeper understanding of that. I mean, evolutionary biologists right now, they've figured out how to look at phylogenies between genes, but they haven't thought about phylogenies between cell types or how that can be traced back to the primordial first cell type or the first organism with two cell types. And this is not health related, but I do think we're going to make amazing discoveries about our collective history of life on Earth through the work we're doing here.
That sparked a hope that I have that will come out of this body of work, which is what are the principles that underlie life?
Great, yeah.
I think a lot of people are often lean towards it's too complex, biology is too complex, it's not it doesn't have the same elegance of physics or whatever it might be. But I think it's just too complex for us to appreciate right now. But I think there's probably some deep elegance and simplicity in what drives life that we just can't appreciate right now.
Amazing. Round of applause for Priscilla and Steve.